Hi everyone, and welcome to the next webinar in our Plantae Webinar series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you are experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you have questions for our panelists during today's webinar, please let us know about those using the GoToWebinar chat box. We will moderate these questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists, powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today to support this series and use the discount code webinar10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. If you are having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with the associated materials and a continued discussion within a few days. Be sure to follow us on social media. The hashtag for this series is #PlantaeWebinar. Thanks again for joining us today. I'm now going to turn it over to my colleague, Jennifer Regala, who is the managing editor for ASPB, to talk about the Assistant Features Editor program and to introduce our panelists.
Hi everyone, welcome. We're so glad that you're here for this webinar today. Thank you for joining us. And I'm sharing my webcam now, sorry. We're very proud of our Assistant Features Editor program. This program exists for both of our journals, the Plant Cell and Plant Physiology. We started this program in 2018 for both journals. Our Assistant Features Editors serve 2-year terms, and they're responsible for a multitude of responsibilities for the journals, including but not limited to what you see right here. The really unique part of this program too is that our AFEs are full members of our editorial boards. They're invited to all full editorial board meetings, trainings, and conversations. There are going to be application opportunities for anybody who is interested this fall for both journals. If you would like more information in advance of our official announcement of the process, please reach out to me. My email's right here, and I'm also on Twitter. That's another good way to contact me as well. Thank you so much for being here today, and thank you for your support of our programs. And now I will pass the microphone over to Mary Williams, my colleague.
Hi there. I'm Mary Williams, and Jennifer and I will be moderating the questions. So please put questions in your chat box. I want to briefly tell you how we're going to organize this panel discussion. We have divided this big question up into a series of sub-questions, and each panelist will be addressing one of these questions. We're going to give each of them 3 minutes to talk about their ideas. Then we'll have a brief discussion within the panel, and during that time, I'll be moderating questions and I'll read out some of your questions for the panelists. So please take good use of the question box, and you're all muted, but I'll be reading out the good questions. So let's see if you can get a shout out. Now what I would like to do is we'll turn on all of the panelists' webcams, and I'll ask them to give a 1-sentence introduction of who they are, and then I'll turn it over to Andrew, who originated this idea for this discussion, and he can give us a bit of the backstory of how we decided to do this. So Katie, if you could turn on the panelists' webcams, I'll ask them to introduce themselves in the order in which they'll be speaking. So we'll start with Lei once we get her going up. So Lei, could you introduce yourself?
Okay. Hi everyone. It's my great honor to join the webinar hosted by ASPB. My name is Lei. I'm a senior editor in Nature Plant from the Nature family. I joined the journal in 2016, and we are a professional editor on Only Journal. And yeah, it's really happy to be here.
Excellent. Arren?
Hi everyone. I'm Arren. I'm a PI at the Max Planck Institute in Golm. I study synthetic and system metabolism, trying to reveal design principles in metabolism and harness them for the good of humanity.
Magda?
Hi, my name is Magdalena Jułkowska. I'm still a postdoc in KAUST in Saudi Arabia, about to start my own group, and I'm working on how salt is changing the plant architecture and how that affects salt plant tolerance.
Excellent. Scott?
Hi, I'm Scott Hayes. I'm working as a postdoctoral fellow in the Testering group in Wageningen. My work mostly focuses on temperature responses in plants, particularly with respect to root development.
Elisa?
Hi everyone, my name is Elisa Dell'Aglio. I'm a postdoc at Lyon in France. I am a plant biochemist, and I work on plant stress responses to biotic and abiotic stresses, and more recently also on epigenetic. So I'm very excited to be here today as well.
And I'd like to mention that Magda, Scott, and Elisa are assistant features editors for the journal Plant Physiology as well. And now, Andrew, would you introduce yourself and tell us something about why we're having this conversation?
Well, by way of introduction, I'm Andrew Hansen, and I'm a professor at University of Florida in the Horticultural Sciences Department, and I'm basically a metabolic biochemist, metabolic engineer, plants to some extent microbes, and I'm very interested now in synthetic biology. So I'll give a little bit of background very briefly on how this got started. Back in mid-March, at the beginning of the lockdown period for most of us, Jennifer and Mary, who you've just met, and editor-in-chief of Plant Physiology, Mike Black, invited editors of Plant Physiology to host workshops initially just for assistant features editors on a topic of their choice. And I proposed the topic, briefly could summarize it as what makes work really pioneering. My actual words were a little longer. It was how to spot a study where pioneering minds have truly leapt a logical gap and created new knowledge that alters how everyone will think. And that's opposed to the standard daily worthy everyday science that we do, most of us do most of the time. So it's in distinction to that. And I want to just show you, and Mary will send you a little more information about this in a tweet later on. But I just want to share before we start 2 classical books that have addressed this issue. This is the more famous by the philosopher Thomas Kuhn. It was in the early 60s, called The Structure of Scientific Revolutions. And Thomas Kuhn is the person we get the idea of paradigm shift from. He's made that part of our vocabulary. The other one is tougher going, but I would say it's even more interesting, by Michael Polanyi. That's a 50s book. Polanyi was a brilliant physical chemist before he moved to the UK from Europe in the 30s, and then he started a new career, actually first as a chemist and later as a philosopher. So he's been in both camps. Heavy read, but really worthwhile. And I just want to quote before I hand back a couple of things from Polanyi, that his book, that really to me hit the nail on the head. One of them is really important for this sort of workshop today, and that's the appreciation of scientific value merges into the capacity for discovering it. In other words, the more you can recognize what scientific value is, the more likely you are to add to it yourself. So this is not just about learning to recognize things, it's learning to do it yourself. And the other quote is, the dilution of the meritorious by floods of triviality makes the recognition of true scientific value particularly difficult. And anybody who's ever been an editor will say amen to that. Okay, so back to you, Mary.
All right, thank you, Andrew. So now we'll move into the sort of formal part of the group, and we're going to start with Lei, who's going to give us her thoughts on this big question. You ready?
Yeah, thanks. Thanks, Mary. Thanks, Andrew. So yeah, we kind of have different roles, like by introducing different topics related to what makes a paper really pioneering. Obviously, I don't think I have a perfect judgment. I'm here to share some of my thoughts. I think a pioneering paper must be based on pioneering research that is rigorous and exciting enough to change how other people think about a scientific issue. As I'm an editor in Nature Plants, I can only introduce our criteria. We have 3 criteria when assessing a paper: the novelty, general interest, and solidity. So a lot of the other panelists will actually talk about the novelty part in a few minutes. And I will be more focused on the solidity and general interest of a paper that makes it really pioneering. I would say most solid papers are not pioneering, but all pioneering papers must be solid. We cannot assume every publication is sound and reliable. A recent attempt to reproduce, I remember it's like 21 or 20 social science papers published in Science and Nature were able to reproduce only 62% of the findings. And it was suggested life science papers only have even lower rate of reproducibility. Some of you may already notice that many novel papers actually suffer from overinterpretation and other mistakes. We know mistakes happen in all kinds of professions, and in science they could be honest errors, but these are not big problems. They are subject to queries and rectification. But we cannot ignore some serious misconduct like fabrication, falsification, or plagiarism. If you still remember the case of a very famous French plant biologist whose initial was OV, studied RNA biology, who published more than 200 peer-reviewed papers and over a dozen received 1,000+ citations. Until 2019, 8 papers were retracted, 5 others received expression of concerns, and 25 others have been corrected. These are definitely not pioneering papers, although some of them were once considered to be. We may also recall some research history of the famous plant auxin binding protein called ABP1, auxin binding protein 1, whose mutants, including TDA knockout, were studied for more than 20 years, with hundreds of papers published in many journals and by many groups. The function of this protein was considered to be essential in plants until 2015, when a CRISPR-Cas a technology was introduced to generate a knockout mutant of ABP1. But this mutant does show any detectable phenotype compared to the wild type control. This is some of the ABP1 studies were considered as pioneering, of course, those papers. But later on, we know there are mistakes there, and we should correct all these things. Of course, bringing up these cases again is not trying to scare any of you, but I want to remind that all of us, scientific rigor and reproducibility is the foundation of a pioneering paper. And we should all recognize that papers are not only a list in our resume, they are also the main component of scientific record. And reliability is the key of anything. I might have spent too much time talking about solidity and reproducibility. Another point I would like to cover is the general interest. So one key criteria of editors like me to think about the impact of the paper is how many other researchers will do their research differently as a result of this published work. It is actually a prediction. We have to predict how many people, or the general interest. And predictions are not always right. I'm very lucky to I'm surrounded by a group of very talented editors, and my editorial members, colleagues, are very kind and friendly to help each other. So a lot of times we act as representatives from different sub-areas of plant research, and if the majority of us or all of us likes one paper, it is likely that this is a paper represents some kind of general interest. Our consideration is not only how many people would like to see this paper right now, but also about how many want to check it, work on it as a kind of basis and also benefit from it in the future. Pioneering papers may or may not have immediate impact, but the impact must be long-lasting and expanding. That's an important criteria of general interest. One well-known case is probably the discovery of Transposon by Barbara Mcclintock. The discovery was made in maize in 1940s and 50s, but it was not well recognized until the 70s, when many other scientists confirmed the mechanisms that Mcclintock found in maize and applied those in many other systems. Because of this pioneering work, Mcclintock was awarded the Nobel Prize in 1983, as some of us already know. So we know sometimes a really pioneering work or paper needs years or decades to be understood and recognized. I hope we will have the wisdom to make precise predictions. Since we are kind of on the front line to read papers and screen papers, like which are the ones which we really think are pioneering and exciting. But right now, what we can do is to keep predicting. I think those are my thoughts. Thank you very much.
Excellent. Any comments or questions from the panelists? Yep.
Maybe I will start. So it was very interesting. Thank you. It's always nice to hear it from the point of view of the opponent, that he is from the editor and not from the scientist who sent it to the editor. I want to focus for a second on one thing that you said about overinterpretation. And I want to challenge you with that for a second. Mm-hmm. So a good friend of mine, Dan Taufik from the Weizmann Institute, has once told me, and since then I see it all the time, that in nowadays paper, it's almost impossible to actually do even simple interpretation, because the editor or the reviewer will attack you for any kind of suggestion you will bring, which is not directly come out of the data. And this does not enable us to communicate a lot of our ideas, which might not be completely solid at this stage, but might bring other people to think. And also, you know, if you look at paper for the 70s and 80s, when things were more less strict than today, you see much more work in which the scientists were able to overinterpret a bit the result, or suggest something which was not completely solid but would make the community think, and it was actually plays a huge role in future development of the field. And today, because we are checked so harshly by our fellow reviewers and editors, we cannot put forward ideas as easily as we can do before. So I just wonder what you think about that.
Thank you, Arren. That's a very well. That's a brilliant question. I would say yes. I do feel that there are a lot of, as you said, overinterpretation is probably. I would say that the first things is like how do we define overinterpretation, right? I don't think it's a problem when you actually look at the scientific problem from a bigger scale, like how it may relate it to daily life of other people, how it may relate it to the strategy of, like, for example, the global sustainability or anything related. It's just how you would like to state it. So, like for example, in the paper, we have a lot of different sections, result discussion. I think in the result section, you have to be very solid, especially when you talk about your conclusions, like you said, simple interpretation. But in the discussion, it's actually you're allowed to discuss a bit beyond the scope of the current study. You can talk about the future application or the potential relevance of your study to other fields, or maybe to a broader scale. I think that's totally fine, and other people would benefit from this helpful discussion. And these are supposed to be speculated. Some of them can be speculated, but it should be in the discussion rather than you have to say anything. This is for sure. This is confirmed. This is kind of, you know, tested or supported as in your result part. I think that's the beauty of discussion.
We've already run over our allotted time for this topic, and there's a million great questions. But what I'm going to do is I'm going to go on to Arren, and I've got all these questions marked, and hopefully we'll get time at the end for many of them. Oh, Arren, you're up.
Okay, sounds good.
Yeah, okay, thank you. So when I thought about what I'm going to talk about regarding pioneering studies, I realized that for me, really pioneering studies always raise the question of why didn't I think of that myself? So this rather simple question actually hides many different aspects. So first, if I ask why didn't I think of that myself, it means that I'm already quite an expert in the field. I think it's mandatory for one to be an expert in the field to be able to identify truly pioneering studies. Otherwise, you might think you are identified like a groundbreaking story, but in fact, you might have been fooled by pompous words or a high impact factor, which happened to all of us. Second, for me to ask why didn't I think of that myself, a study must hold some unforeseeable aspect, which is not a direct logical step from past knowledge or experiments. This could be related to a methodology not previously used, an experiment not previously suggested, a new unifying theory, or simply results which are completely unexpected. However, despite the unforeseeable aspect of a study, it should be simple enough such that I could truly ask myself, why didn't I think of that myself? So I want to argue that most of the really pioneering studies are simple enough to be understood almost immediately by people from the field, and hence raise the feeling that they could have thought of it themselves. And I want to give one such example from my field of synthetic biology, or more precisely synthetic metabolism. When I read more than a decade ago the first study that described the introduction of a synthetic photorespiration route into a plant in order to boost carbon fixation rate and photosynthetic yield, the first thought that came into my mind was, indeed, why didn't I think of that myself? So I was already quite an expert in the field of cellular metabolism and metabolic engineering, so I could realize that this is truly a pioneering study. It was unforeseeable in the sense that no one has previously thought of using such a synthetic metabolism, metabolic engineering approach to redesign this key process. But in retrospect, it was so simple conceptually that I really could not understand why I did not think about it myself. After all, applying metabolic design principle to suggest, analyze, or implement alternative pathway to re-assimilate glycolate back into the Kelvin cycle is quite straightforward once you think of it. And indeed, as expected from a really pioneering study, this work of rerouting plant photorespiration had led many others, including myself, to pursue such an approach. And today one can see multiple fascinating stories being published about different possible pathways that are tested to enable a more efficient glycolate metabolism, supporting better or higher photosynthetic rate. So I guess that for many, not just for myself, when they read this original paper, the first question was, why didn't I think of it myself? Yeah, that's it. Thank you.
Excellent. Any comments from the panel for Arren? Yeah, Andrew?
Just to emphasize something that Arren said about how it's necessary to have a deep knowledge in the field before you can really recognize something. I just want to stress that deep knowledge doesn't equate with many decades spent in the field, because it's almost sometimes the opposite, that somebody comes in with a very sharp focus and looks hard at the field and understands it deeply. You can do that in months, not decades. And some of the most, I would say, amazing people that I know in science, I don't know him personally, but Ron Milo, who Arren knows very well, that is his modus operandi, I would say. Would you comment on that, Arren?
Yeah, I completely agree. You definitely don't necessarily have to have, your white beard to be an expert in a field. If you come, to come to a new field and you really dedicate to yourself to actually understand it from, from the bottom up, from the basic details, rather than just from, from above, then you can become an expert in, yes, I guess in a year you will be able to contribute and you will be able to assess pioneering works. The problem is still, Andrew, that many people do see themselves as experts in field that they are not necessarily expert. And this leads, in many cases, for some works and study to gain much more attraction as pioneering, despite the fact that, you know, there might not be that pioneering. And I think that's also maybe one of the major challenges for editors, because editor cannot be an expert in all of the different field that is viewing the papers in. And there are, you know, researchers that are better than others in selling the work by using, as I said, pompous words and, you know, stuff like that. And it's quite difficult to be able to understand which of them is really at the core pioneering unless you have an intimate knowledge.
Excellent. I'm going to ask one quick question, and I'll let one of you pick to answer it. Truly pioneering work is often high risk, and it can be very difficult to get funding or to convince others that it's worth doing. Any advice? Anyone want to take that challenge? Yeah, Arren?
If I'm talking too much, that's meant me to shut up. But I will just say that in my experience, truly pioneering work are actually easier to finance because, at least in Europe, I'm not talking about the US. The US might be completely different, but in the European kind of sphere, they really want to see when you are applied that it's not more of the same. And if you can convince them that what you are doing is not more of the same, and I'm talking about both national, in my case, in Germany, but also within the EU, if you convince them that what you're doing is really pioneering, they will enable you to take risks that otherwise they will not let you take. So I think that the funding agency are actually behaving, from my personal experience, are actually okay with that.
All right, I'm going to move on. Thank you for that. Now we're to Magda.
Yeah, all right, cool. So I think like an important part of a really truly pioneering work is in the unconventional data analysis. And from my perspective, data analysis is where magic happens, where we forge the data into knowledge. And it can be at so many levels, because data analysis and data visualization is also part of data analysis. So one very simple example is from the beginning of 20th century, where there was a cholera outbreak in London, and people were trying to understand where is it coming from. And there was this person called John Snow, where he plotted the numbers of the cases on the map. And that allowed him to kind of pinpoint where the hotspots of the outbreaks were coming from. And it's very relevant, and we're still using this kind of methods at the moment as we speak. And it's something very simple as combining numbers, tables with something very visual. Right now, there are also other ways that we're using data visualization that are really enriching our understanding. Examples are, for example, the circus plots that people are using in subgenome visualizations, which are providing really an increased insight of how genomes or subgenomes are evolving. And that brings us also to the sequencing. So back 20 years ago, the costs of sequencing were super high. Right now they are plummeting, and that gives rise to computational genomics, which allows us to basically not do even a single sequencing ourselves, but just pull all of those sequencing from online and do the analysis depending on what we're interested in. Is it like a specific gene evolution or a genome evolution in general, or even the evolution of specific pathways, either signaling or biosynthetic pathways. And also those costs are, the costs of sequencing are also allowing us to compare not only variation between the different organisms, but within the organisms, allowing approaches such as genome-wide association. And I think with that, what is really interesting in being plant biologist, the models for genome-wide association that are being used in humans to pinpoint specific genes responsible for diseases or other phenotypes that we're interested in. Is there a gene for being good in music, or is there a gene for, I don't know, being very entrepreneurial? Those studies are also sometimes pseudoscience, so we have to be very, very careful of what exactly are we using, and we have to be also very, very I think, aware of what is the context of the data. What can you do with this kind of meta-analysis, and what can you not? Because it's very, very prone of, if you don't know what you're doing and if you don't understand the context of your data and how you should normalize for different factors, it can be very dangerous. So I think one point that I would like to highlight is that those meta-analyses should be always taken as a hypothesis generating approaches rather than proving something. The fact that something is associated with whatever you're interested in is not a proof that it is. It's just an indication. And another type of like data analysis is from pictures. So I'm working myself on phenotyping, and if you've got a picture of the plants, there are so many ways of analyzing that picture and combining that with everything we know about, for example, water transport. So right now we can use the knowledge of plant cross-section with the knowledge that we know about the individual parts of the plant that are permeable to water, and what are the dynamics. We can set up a model of what we think that a good anatomy of a plant is for the most efficient water, for example. And those, they're saying that there is not a single model that is perfect, but a couple of models are very useful. And I think there's also a movement in including more and more machine learning in those analyses. So that's kind of beyond human intelligence. But another trend that I wanted to mention is so-called latent phenotyping, which is the fact that we can measure something or we can recognize that the plant has, 10 leaves. It's also very biased of how we look at plants. And what latent phenotyping is doing is kind of combining the genotyping information with everything what you can measure about a picture of a plant and kind of finding a way where is the strongest signal between them. Yes, and with that, I think the collecting the metadata properly so that it's representative of what you're interested in and reproducible. And Lele was mentioning earlier that reproducibility is one of the very important factor. But I think reproducibility, it's a result of how well you're documenting all of your conditions. Because if you're using slightly different lights, slightly different growth chambers, all of us know that your plants are just going to grow a little bit different. And I think data reproducibility is going to become more and more important. That's it.
Comments or questions from the panel? Yeah, Scott?
I really like your example about John Snow's plotting cholera outbreaks. But I think it kind of links to what Arren was saying of this, why didn't I think of that earlier? It's something which I think pioneering work, it really sometimes, it seems very simple, but I mean, it's obviously not because no one's done it before. I think that's for me, one of the main points of it is just how simple it seems, but in reality it's not.
All right, I have a question from the audience, which is, how important do you think new technologies and approaches are to making something into a pioneering work? And is it less likely to be pioneering if you use sort of standard techniques? What do you think?
I think the new approaches are definitely very exciting, but I think there is still need of linking it to a biological function. So you still need an expertise in the process that you're that you're looking at. I know that Arren wanted to say something as well.
I'm not sure if you have time. If you have time, I'll just say one comment. I think that definitely new technology can lead to pioneering works, but I don't think it's mandatory at all. I think that a lot of the really pioneering works were just using completely known methodologies. They just thought about it in a different way to approach it. So it's definitely not mandatory. It's helpful, but it's definitely not mandatory.
Yeah.
I also have one comment. Like new approaches, like what I can see is like the importance is whether you would consider it outperforms the conventional approaches, and also whether you can use it to find something really novel that old conventional approaches cannot capture. I think that's the most important part of using the approach.
Yeah, totally agree with that, because when you're doing all of those machine learning, and machine learning is going to tell you photosynthesis is important in plant growth. Well, they've known that.
Yeah.
Excellent. One quick comment from the audience, which is totally agree with that data analysis is the way to discover new reality in science. People should think creatively for analysis rather than stick it into pipelines. I think it's quite good. All right, we're quite, tight on time, so I think it's time to turn it over to Scott now for your topic.
Okay, I'll keep it relatively brief. So my subject is going to be about how combining different subjects can really lead to new insights in work. So I suspect that many of you have heard the expression "standing on the shoulders of giants." And this is a popular cliche about the scientific process, and it kind of nods to the idea that science is incremental. All science is simply tiny extensions on the massive body of work that we have existing. And well, I think there is some truth to this. I think actually a better metaphor for the scientific process would be building a house of cards. So if we can imagine a house of cards is the entire total scientific knowledge in any given field. As we build this pyramid, we can imagine that if we lay two cards together, this is an individual study. Now, these individual studies are, they're inherently unstable. Some of them will fall while others remain standing. For me, one of the most interesting and important parts of science is the papers which link these studies. So we can imagine this in our metaphor as the cards which bridge between these two, as this brings more stability to the overall structure of the pyramid. What's more, these cards actually form the basis for the next layer. So they actually kind of promote our moving on through the science and actually kind of going towards moving on with upwards and really improving our understanding going forward. In addition to this, we also see a different kind of study. This kind of study bridges not between concepts within a field, but between concepts between entirely separate fields. In our metaphor, we can imagine this as there being two pyramids and having a card between these two pyramids. When this happens, this bridging card actually has the ability to form an entirely new pyramid itself, and a new scientific discipline can be born from this. For me, one of the best examples for this historically is the founding of the field of molecular biology. So if you look at one of the key papers in molecular biology, defining the structure of DNA, this is really it was based on a combination of physics, genetics, biochemistry, and crystallography. And yeah, it really just led to a massive revolution in biology, founding extremely diverse fields such as biotechnology and population genetics. So I would argue that the key to the success of this kind of founding paper is a willingness to look outside of a single field and to draw inspiration from multiple sources. Of course, incremental studies are often the source of this inspiration, but for me, it's the studies that link these concepts which can be considered truly pioneering.
That's lovely. Panelists?
Elisa?
Sorry, thank you. I just have one question about it. The fact that you say when you have two pyramids and you try to merge them together, one problem with that is that you don't have an audience. Because if you increment knowledge in a field, you have all the experts in the field that will read your paper. But when you try to bridge two different fields, it's more difficult to make yourself heard. So what would be your advice for someone who tries to do that?
Yeah, I guess it's trying to, well, I mean, if it's pioneering enough, it will create its own audience, right? I mean, if it's really something which, I mean, this is the kind of, I think it's going to tie in with a lot of what other people are saying, this kind of, I think you can kind of appreciate this kind of beauty in doing something which is seemingly simple, but it's actually very difficult to do, that kind of thing. I mean, it's, yeah. I guess it kind of comes in the longer run if it is.
Yeah, it takes maybe more time.
Yeah.
But in the end, we succeed.
Magda?
Yeah, I was just wondering, because your metaphor of house of cards, for me, it's something very fragile. Did you want to comment on that? Was it on purpose?
Yeah,
or?
I consider this, and actually I think it's kind of apt as well, right? I mean, the idea that we are certain of anything in science is very misguided. And inevitably, if you make these new pyramids, sometimes they can disrupt the understanding that we have about another field. The molecular biology is a case in point for biology. It really changed the way we understood biology completely.
Andrew, did you have a comment?
Yeah, just a sort of practical comment, maybe at a relatively low level bridging between fields, to respond to the question. One practical thing you can do is to make sure you understand the vocabulary in the other field that you're trying to bridge to well enough that the people in that field are going to recognize that you're not talking nonsense. And the big aim in doing that is having an ongoing dialogue with people from other fields before you presume to trample on theirs, because we're all in tribes and none of us like the people from the other tribes. I mean, there's a built-in out group type thing, and you have to be able to overcome that. And vocabulary and not making a hash of using other people's, other disciplines, language and thought process, that's a thing to avoid, at least. Yeah.
Those are very wise words. Let's move on to Elisa.
Okay. So now I have 3 minutes to convince you of the importance of missing pieces in science. But obviously, in order to recognize the importance of a missing piece, you need first to have the rest of the jigsaw, or at least enough of it to spot what is missing. In chemistry, the most famous jigsaw is, of course, the periodic table, invented in the 19th century by Mendeleev, and that immediately showed 2 potentials. The one of putting some order in all the known elements on the basis of their proton number, and the one of predicting the existence of undiscovered elements with predictable reactivity. Nevertheless, when radioactive elements were discovered or synthesized a few years later, that was not just a fic in an empty box. That was the start of a new era for human history, made of new energy resources, wars, and medical advancements. It's a bit the same feeling that we perceived when in 2012, the CERN announced the discovery of the Higgs boson, a long-sought particle that was hypothesized for the first time in 1964 by Peter Higgs. Okay, Peter, you were right. Thank you very much, nerd. And so what? Well, now we can finally explain the force of gravity and attempt new predictions on the destiny of the universe. I guess this is all quite clear now, but what about plants? This may all seem quite abstract for a plant biologist. After all, biology never had a periodic table or a standard model, nothing like a frame where to add pieces. But I believe that the concept applies here as well. If we think about the history of life as our standard model, and those pieces as the pieces of this puzzle, then it all starts to make some sense. Adding a missing piece in biology can be the discovery of a new or ancient species, the creation of a new species in the lab, and even the discovery of a molecular mechanism that helps us connecting the dots. I will just give you one example. Charles Darwin used to say that the origin of flowers, and here I have a flower, is an abominable mystery of evolution, an invention so recent and so successful, but apparently so complex to achieve. Now, thanks to remarkable comparative molecular biology work, we know that the gymnosperm of Welwitschia mirabilis makes rudimental flowers too, with sterile ovals as well. And this because of a cascade of transcriptional activation that is equivalent to the one of angiosperms, suggesting that all the elements needed to construct the flower apparatus were already present in the common ancestors of angiosperms and gymnosperms, ready to bloom and surprise us with amazing colors and shapes. Another piece found its place in the jigsaw of life. Thank you. Yes, Arren?
So fascinating. I guess I might want to argue about the notion that we don't have unifying theories and models or structure. So yeah, we don't have something as beautiful as the periodic table, that's true, or the standard model of particles and so on of physics. But we do have some idea which are at the core of what we are doing, all of us as biologists in different fields, starting from the starting concept of DNA, RNA, protein, or with the notion of evolution, that nothing makes sense unless in the light of evolution. And also, of course, the story of plants is interrelated directly for the story of insects or pollinating the plants and the flowers and so on. So I think that we do have some generalized theory that are true for all of us in the life sciences that cannot be presented so nicely, maybe like in chemistry or physics, but are still as important and as at the core as in our case, as in the other fields.
Yes, I totally agree. There are also many other examples that I could have given. For example, also metabolic pathways are a way to structure what we know about how cells work. But of course, it's not like in chemistry or in physics where everything is so beautifully described now. But we are going there, I think.
We have some excellent questions.
So let's say another word that I'm not sure we are going there. Oh, sorry. One more. I'm not sure we are going there because I think that biology, by its essence, it's much more complex than chemistry and physics.
Oh, yeah.
So I don't think that we'll ever have something which is as simple as they can put their stuff on the board. So for us, it's always going to be much more complicated, and hence the pioneering studies for us are also going to be much more diverse than in their case. Sorry for.
Yeah. This means more pioneering study, which is good.
Yeah, I think someone said it's not exactly rocket science what we're doing. It's much more complicated.
All right, let's turn it over to Andrew, and I'll have a few great questions for you at the end.
Okay.
Yeah, I want to defend the viewpoint that really pioneering work will be in some sense beautiful. Now, I want to start with philosophy, because philosophy calls truth, beauty, and goodness, it calls them transcendentals, which means rising above. And that's because they're our highest human values, truth, beauty, goodness, and they're in some way unified because wherever there's truth, there's beauty and there's goodness too. And yet all of them are hard to define objectively. So we have to admit that recognizing truth, beauty, and goodness means making subjective, emotionally based value judgments. So that much is admitted by everybody, I think. So in what sense can an advance in science which so aspires to be objective and rational, in what sense can you call it beautiful if recognizing beauty is a subjective process that involves the emotions? And there are two classical answers to that, and I'll give them to you in order. First, science that alters how we think about reality isn't an objective mechanical business. Everyday science, the worthy everyday science, is objective. It's mechanical, it's incremental, as others have said. It logically follows prevailing paradigms and it uses familiar methods. That's everyday science, but pioneering science is completely different. It makes these leaps of creative imagination that may come before experimental work and that require this deep understanding and commitment to and even passion for a field. People immerse themselves in these fields. And notice there that I'm using words to describe this kind of science like creative, imagination, commitment, passion. And they're all emotion words. They're not cold logic words. So if doing pioneering science in the first place is harnessing the emotions, shouldn't we expect recognizing it to do the same? So I think, exhilaration and delight even are totally fitting and maybe essential responses to an encounter with pioneering science. It should delight you, thrill you. Now, the second classical answer is that it's the short one is that you can't, since we can't really separate beauty from truth, then what's scientifically true is necessarily beautiful. But you can go a bit deeper than that, because in fields beyond science, our concepts of beauty take in simplicity, symmetry, harmony, elegance, power, and they aspire to be universal and timeless. So exactly the same applies to the concept of beauty in science. All those words apply to something that you would really be captivated by in science. And to end, I'm going to add a little bit controversial, and it goes back to the transcendental of goodness. Now, to some degree, what's good can be equated with what's useful. So for me, pioneering science that's potentially useful becomes more beautiful. And that's why I see beauty in synthetic biology, because synthetic biology looks back at biology with fresh eyes and says, it didn't have to be this way. In other words, biology took a particular path, but it could have taken a lot of others. But synthetic biology also looks forward in hope and says, it doesn't have to be this way in the future. And then it sets about exploring solutions that nature hasn't, finding good ones, you know, to apply to the world's problems. So that to me is a form of beauty. That's it.
Excellent. Panelists? Yep, Arren first, then Magda.
So it was fascinating, Andrew, but I must admit that in one place I don't agree with you, and I want to say it in front of everyone. I don't think that useful is beautiful, and I don't think that useful is good. Actually, doing installation when my sync logging is very useful, it's not beautiful. And you know, and I think that many of the papers that we see today are very, very useful, but they are very far from being pioneering, and they are also very far from being beautiful. So I find it very difficult to put the word useful into the equation in this case.
So let me respond briefly to that. I said that utility was an added aspect of beauty. It wasn't required for it, so that it could make something, at least in my eyes, and I recognize that's controversial, it can make it more beautiful. But I totally agree that beauty in science does not require utility, but I would argue that it's certainly not incompatible with utility, and which some people do argue. You know, the more useful it is, the less interesting and beautiful it is, would say. But that would be a very classic position. I don't take that. I think utility can increase the beauty of science, and it's certainly, at the very least, it's neutral. And I think you'd agree with that, right? It doesn't detract from it necessarily, but I said that to spark some argument, because I know not everybody thinks like that. Magda.
Yeah, so I wanted to poke your brain a little bit about the beauty of science, and I usually, the papers that I find beautiful are very mechanistic. Do you think there is, are there any examples of works that are exploratory and beautiful?
Exploratory. Can you help me with that word?
So more hypothesis generating, as in rather than hypothesis testing? Do we need a full mechanism to have a work that is truly beautiful?
Well, I think that the kind of beauty I'm talking about involves a synthesis of some kind. It doesn't really involve a new idea, which may or may not be true. It involves some bringing together, some harmony, some symmetry, some recognition of order, of a new power.
Okay.
They.
Oh, hi. Yeah, Andrew, I would like to ask you a question, like the beauty of a paper, like we are talking about pioneering paper. How much would you attribute the beauty to the beauty of the science itself, and how much would you like to attribute the beauty to the beauty of writing?
Mary is going to address the beauty of writing, but I think it's practically unclear inward-looking writing is a sure way to minimize your chances of being recognized as beautiful.
All right, I'll go ahead then. We've really only got 5 minutes left, so I'll be extremely brief. This is Mary again, and we can only show 6 webcams, so that's fine. But I was asked to talk about storytelling and its role in pioneering work. Uh-oh, now I have to do something with my computer. Oh dear. Oh, here I am. Okay, so I think Leigh mentioned Barbara Mcclintock, and this is someone that we all think of when we think of a super genius. She's been described to Einstein and John Nash and Newton, and yet her work was not considered pioneering at the time. It was considered meaningless or incomprehensible. And it wasn't until other people interpreted the same work and you know, replications of the work that people realized the genius of her work. And so, as Andrew said, you can't have work that's really going to revolutionize the field if you can't explain it to people. And so, of course, you know that I'm really big on people learning how to be effective communicators. And one example that I wanted to give you, I'm going to have to read it, is comes from a paper that Andrew wrote. It was actually a review article, and he was talking about the you know, the kind of exciting things that are happening in synthetic biology. And he wrote a review article in Annual Reviews called "Metabolite Defense and Metabolite Damage Control in Plants." Sorry, metabolite damage. And he described metabolism in plants. He said, metabolic pathways are thus narrow, well-lit tunnels of known chemistry boring through a huge, dark matrix of unknown chemistry. And to me, that was a really radical idea. And the way he described it, basically painting a picture, helped me to understand a concept that was completely new to me. So I would argue that if you have a pioneering idea, you need to figure out a good analogy and a good way of explaining it to people who aren't light years ahead of you or on the same wavelength as you even. So please learn to communicate effectively, come up with analogies, and I have a visual thing here. Oh, where is it?
Yeah.
Yeah. So this is one of the most famous figures of all time, and it's Darwin's "I think" and a little sketch. If you can sketch your ideas, you can communicate them. So I'm going to turn my camera off again because I have too many windows open. But I have a couple of just brief comments from our massive number of questions. Thank you so much, everyone. And we will be addressing your questions in the Plantae network. But here are some of the things people have been talking about. Someone said, read more. The more you read, the more able you will be to embrace and move into pioneering research. Someone asked for recent examples. They said many of the examples you're giving are kind of old. Does anyone have any recent examples? Someone said,
we
can
don't
a
want to
review...
go into politics.
Sorry?
We don't want to go into politics, so it's better
no,
saying, you know,
maybe in our written
No,
text.
I think it should be said that we did decide, as Arren says, to deliberately not talk about recent examples of research. That was a deliberate choice.
Another comment was the bias in science, not only bias between disciplines, and Andrew mentioned, you know, learning your tribe and learning to communicate with other tribes, but everyone knows that there are biases in where the country of origin of the research is or the gender of the people. So that is something that people are quite concerned about in the comments and discussion. And several people said, and maybe this is a good place to wrap up, if you could all give your thoughts on this as we close. What advice do you have for scientists? How can our audience of more than 600 people think about what they can do to start to, you know, maybe move their research into a pioneering direction? So what is your advice for early career scientists and others who maybe want to be pioneers? And I think we'll go in the same order as we spoke in. So maybe Leigh, would you be willing to start?
Okay, yeah, I'll be happy to. So I think the first suggestion, read more, is definitely a key. You have to read more. You have to know the big picture of your science, and then position your study into an important direction that your field is pursuing to. That's my suggestion.
Great. Arren?
So building on top of that, because I completely agree with Leigh, of course, here, but I would actually suggest something which sounds contradictory. I would say do not focus on trying to be pioneer. Do what you do the best you can in you know, putting all of your heart, all of your knowledge into it, and you'll get there. Because if you will be obsessed all the time of how I get in pioneering, that's not the right way. The right way is simply do what you're doing, and you'll get there.
Magda?
I think talking to people, and more importantly, listening to people is very important, especially like if you're in the environment of even your own university. Go through your corridor and talk to all of the people working even at something, other topics that they are working, because their collaborations may be hiding there. And also I really like talking to students that are just starting their internship with us, because their naive view can be very enriching.
Thank you. Scott?
I would say do what excites you. If you want to make these collaborations with other people, sometimes it can be hard going because, well, as Andrew made the point, you're speaking a different language often. So if you do something that excites you, you're going to really push you to try and understand something from another perspective.
Elisa?
Yeah, I wanted to refer on what Andrew said before about beauty of science. And of course, what's beautiful is subjective. But I think if you try to figure out what's beautiful for you and focus on what you really like, then it will be much easier for you to invest yourself in that, and also to tell a good story. Because if you like your story, you're going to tell that story better than another one that you like so, even if it's maybe more pioneering or promising.
Nice. And Andrew?
Wherever the herd is running to, run in the opposite direction.
That was fascinating. To the audience, I just want to let you know that we all have written a short essay that we will be posting along with our video, and I will also be posting your extraordinarily good and plentiful questions, and I will invite the panelists and the rest of you to comment, read the questions, think about it. This was a fascinating discussion. We should have scheduled a 4-hour thing, but I'm sorry, we're finished. And now I'm going to return it to Katie to wrap up. But I would also like to thank all the panelists. This was really inspiring and very, very interesting. So thank you all. And the audience, 600 of them, say thank you very much.
Thank you for organizing.
Thank you. Thank you.
Yeah, thank you all. This has actually been a record-breaking registration and attendance for us today. So I think that really speaks to the value of this topic and the value of our community members like you who've helped us to make this possible. Be sure to join ASPB because that's what supports this webinar series and makes it open to everyone. And also be on the lookout for more online events. If you go to plantae.org, Plantae Presents, we also have upcoming webinars listed there that we would love for you to participate in. And we'll see you next time. The recording of this video will be posted within a couple of days, and we'll be continuing this discussion on Twitter. So follow the hashtag Plantae Webinar, and I'm sure there'll be a lot more lively discussion on there. We'd love to also hear your thoughts and comments. So we'll see you on Twitter and see you next time in our next webinar. Thank you.
Thank you all very much. Brilliant. Loved it.
Bye.